The second assignment for this week is a microscope lab. Now that you've had some general information on microscopes, I'd like you to test your skills using a virtual microscope to view several specimens and identify several professionally done microscope slides. So in order to complete this assignment, you have to be familiar with using the virtual microscope. If you look, you can read through the directions here on your own. Then when you're ready, let's take a look at the questions before we go to the virtual scope lab. The questions are: how many objectives are on the virtual scope? What are the magnifications listed on each objective? And then you'll be looking at each of the slides on the right side of the website and describing what you see. Describe color, shape, distinguishing marks. Tell me what they look like. Make sure you're following the tutorial for how to use the microscope. You should be looking at everything under more than one magnification. So you'll be describing the letter E, the onion root tip, a bacterial capsule, and a cheek smear. Then I'd like you to identify the parts of the microscope that is down here. This microscope here goes with questions 7, 8, 9, and 10. So tell me which letter is with the ocular. Which letter shows the coarse adjustment, which letter shows the fine adjustment, and which letter shows the stage centering. So you're looking at these letters A, B, C, and D to identify those four things. Last but not least, if you scroll down to the bottom of the page, there is a link for the next website, and what I'd like you to do is identify the specimens pictured for letters A through G right here, and that link is right here. So let's take a look at the virtual microscope. In case you don't know, if you right-click on any link, then you can click the instruction that says open the link in a new tab, and then it gives you two tabs to work with. It makes it a lot easier for going back and forth from directions to the actual lab. So the instructional video, if you don't have QuickTime installed on your computer, you can play it in Flash. That'll go through and show you how to use the virtual microscope. We can go right to it.
Welcome. Click here for a tour.
It already starts the tour for you. It will guide you audially as well as visually in using the microscope, but just a couple quick things. I do expect you to go through the tour, and if you need the volume to be turned up a little bit or closed captions, we'll type out whatever it's saying. But just to make sure that you don't forget, over here, this switch view goes through this. The view you're seeing now is looking through the microscope. This view is looking at the microscope. This gives you a helpful checklist about things you need to do whenever you put a slide onto the microscope. So for example, if you turn the light on, you go down here, it's fairly easy. You just click light on, and then it checks it off for you. The rheostat is this knob right here. That's showing you how much light is going through the light source. You choose a slide. The first slide is the letter E, which you will need to describe. To center the specimen, and it says temporarily open the iris, that would be the diaphragm right here, so that you can see the amount of light changes in the middle here when you do that. If you move it all the way to the right, it's showing the most amount of light. And to center the slide, which means you want that black dot to be right above the hole in the stage, you use these two knobs here. And it takes some practice because it's backwards from what you would normally do. So now you can see I've got my specimen centered over the hole in the stage that allows the light through. You want to start the stage at the top position. That means using your coarse adjustment knob right here, or the coarse focus. You want to lift the stage all the way to the top. And then when you go through, first you need to adjust the ocular. You do that by sliding these two until the white circle is just one circle. Then you would use the coarse focus again, and this time you want to go down until you see what you're looking at. You can adjust the diaphragm, see that makes your specimen clearer, And then you need to bring the specimen, in this case it's just the letter E, but you need to bring the specimen back to the very center of your field of view, which is what this circle of light is called. Once you've done that, now you can switch to different objectives. It's best to go in order and to focus each time. So for example, we're on the lowest power. If I switch to the next highest power, it's the one in the back here that you're looking at. And then refocus using the fine adjustment. The only time you use the coarse adjustment knob is on the lowest power, and if you try to use it here, it'll give you a warning saying it's not a good idea to use the coarse focus unless you're on low power. Do you want to continue, yes or no? So of course we want to say no, and then we can focus our letter E a little bit more until it's as clear as we can get it, and you can continue to adjust where this is. The more centered it is, the easier it will be to find it on the next highest power. See, as you can tell, when you go from one power to the next, it gets a little more difficult to see things. Always remember you can adjust the amount of light that's coming through, which does help to let you see what you're looking for. Remember, the top knob here moves it forward and backward. The other knob moves it side to side, and it will be backwards from what you think it should be. And then we can go all the way up to 100 times magnification. Oops, don't want to use the coarse focus. I'd like you to use the controls here on the microscope to view all 4 of these slides and describe them to me with a good amount of description for each one. If you don't want the checklist on, once you've done it once, that's fine. You can always go back to it here if you need to go back to the outside view of the microscope. Simply click switch views. So that's the tutorial. If you don't want to watch it, that's fine. You don't need to. If you want to choose try this, it gives you some puzzles to try and figure out using the microscope over here. And that's this portion of the assignment. And then again, we can go back to our blackboard. Then again, you want to click on this site. And if you want it in a new tab or a new window, you can simply click there. And what I'd like you to do is simply identify the slides that you see throughout this website. It says, study the following images, make note of the descriptions so that you can identify them later. It will take you through a variety of slides. Click the arrow when you're ready to go on to the next one. Make sure you identify in your mind, okay, this slide is showing us a blood sample, red blood cells. Then when you click the next slide, it'll show you dust mite, so on and so forth, and then at the end it has a little quiz. It'll show you the image and you have to identify it. And that's what I'm asking you to put on your paper at the end, or on your assignment. So if you have any issues or questions, please let me know for the second assignment in this week. If you have any extra time this week, it's always good to catch up on the things that you are behind on, and then enjoy your Thanksgiving break.